All right, so we got phototransduction. And simply, I mean, like I said, convert light to electricity, okay, to an action potential. Well, greater potential, action potential, yada, yada, yada. The whole thing is electricity, right? And this is what's going to be done by the photoreceptor. Specifically, it's going to be initiated right here in the outer segment. There is, we could spend literally an hour talking about these three slides. There's just there's a lot of physiology that occurs here with the eyeball. It's not necessarily enough to just have the light hit my retina, get an image to form basically, or get an electricity to form that sends it all the way back to my occipital lobe. But we have on center, we have off center vision, this ability to create nice sharp detail as I'm looking straight ahead here. That's going to trigger certain pigmented cells. Here are rods and cones here in this region. There we go. But then it's also going to cause an inhibition right here in my field of view to start making it not as detailed and making it just a little fuzzy so I can focus on what's coming here straight at me. So there's a ton that goes into vision. We are just going to scratch the itty bitty surface of this thing and what is happening, okay? So we have the photoreceptors, we have our rods and our cones. And we talked about this, I believe, last week, talking about where rods are and where cones are. Cones are in that fovea centralis region, that macula lutea region, right? That's where we have our greatest concentration. As we get further away from that optimal spot of greatest visual acuity, we get further and further away from that, the number of rods dramatically increase and the number of cones decrease. Right? So where we're going to find our greatest visual acuity, our sharpest vision, is going to be in that macula lutea / fovea centralis, where we have our greatest number of cones. When we look at the rod and the cone, the area where we have photopigments is in this outer segment here. Okay, that's going to where it contains what are called disc, and the disc are what actually do the absorption of light. And if I go forward for a quick second, here are these stacks of basically disc. If you've taken 101 before, it almost looks like a thylakoid, really, for photosynthesis and how plants turn light into food energy. It looks almost, if you've taken 101, then you know, I mean, good God almighty, that looks exactly like a granum of thylakoid. So it's kind of weird and creepy. Our eyeball looks exactly like what's going on inside of a plant. So you can have that kind of visual if you know what the hell I'm talking about. If you don't know what the hell I'm talking about, just men in black yourself and just forget it, all right? But here is this outer segment, okay? And here's, shockingly, shaped like a cone. There's a cone. And shockingly, this is rod shaped, so it's called a rod. So here is where these photopigments are going to be really kind of housed in here. And what light is going to be causing those photopigments to change, which initializes an action potential. So if we go back here, all right? So this is where we have our photopigments, okay? Photopsins, rhodopsins, okay? That's what we're talking about here, all right? When you look at the rest of this visual receptor, the outer segment has all the goods, okay? Has all the photopigments that respond to the light. But we have an inner segment, and it houses all the organelles. So it's, doing all the work that any typical cell does. It'll have a nucleus inside of it, and that's the cell body. And then I don't know if I want to, kind of do, kind of don't, probably not. The synaptic terminal contains neurotransmitters. It specifically contains glutamate. At least that's what we believe. Most books say it is believed to house more glutamate. I'm not going to ask. Okay, I won't ask the specific receptor here, or I'm sorry, the specific neurotransmitter. But just know that we have neurotransmitters there. And the interesting thing about glutamate is that, it can cause depolarizations, it can cause hyperpolarization, so it can do a bunch of fun stuff, but we're not going to get into that. Just neurotransmitters, all right? So the light strikes those inner segments, the outer segments, causes those discs filled with photopigments to change shape. And we'll see that here in a second, okay? Once those photopigments become chemically unstable, they trigger the ultimate action potential. They trigger the electrical event that generates the action potential to go all the way back to that occipital lobe. So you can see what in the world is coming at you. Cones, again, concentrated in that fovea centralis, macula lutea. The fovea centralis is in the center of the macula lutea there at the back of the retina. That is where we have our greatest visual acuity. The fovea is like 100% rods. The macula is like 90-something% rods. Or I'm sorry, cones. I can't think. I'm just so lightheaded today. I apologize. Cones, 100% cones in the fovea, 90-some% cones within the macula lutea. And then as we get away from that, as we leave that region of the eyeball in the back of the retina there, then the rods, shoom, huge amounts of those, okay? The great thing about having a huge number of rods, guys, is that they are very sensitive to dim light, okay? Cones don't really function that all that well in dim light. They need bright light to function, okay? Activated by high intensity light, bright light, okay? And these allow color vision where these allow black and white vision, okay? So they're going to give you that when you go down into the basement and you can't find the light switch, rods are the things that are going to be activated. You're in a dark movie theater. Rods are going to be the ones that are activated more than cones, okay? They don't do much for high resolution, okay? They give a very blurry image, okay? Cones are involved in huge amount of visual acuity, okay? That has to deal with some of the physiology that I don't want to get involved with, okay? Just know, rods are more numerous over the entire surface of the retina, sans the macula lutea / fovea centralis. Rods work under very dim light, low levels of light. But because they can function in low levels of light, they are very sensitive. So if there's even just a little bit of light shining in that room, the rods pick it up, cause the change in the photopigment that we'll see here in a second, I believe, on the next slide, and then allow a blurry image to be picked up. Cones, again, concentrated in the fovea or the macula lutea. They function under high intensity light. They give you color vision. There's 3 different types of color vision pigments. And this produces a very, 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 very sharp image, okay? Again, whenever you go to pick something up to read, you automatically put that piece of paper or move your head to get that image to focus on the fovea. These photosynthetic pigments, we are going to call them opsins. We have a rhodopsin in rods, and we'll just call it photopsin in our cones. There's 3, as you can kind of see there. They respond to different wavelengths. So, if you have the genetic disease carried by mom, it's an X-linked genetic disorder. X-linked recessive, I guess, if you want to be specific. That's carried only by the mom. The dad either has color blindness or doesn't have color blindness. The dad never gives it to his son. Dad gives him the Y chromosome. This is something always passed from son, or I'm sorry, from mom to son. Okay. But this is, when we look at our photosensitive pigments, it's basically made up of a protein and a vitamin. Oops, sorry. So we have what are called opsins, and there it is. There it is. Okay, that's the protein part, the structure. And we have a vitamin A molecule attached to it. And that for us is retinol. There are 3 different forms of vitamin A. Retinol is the one that is involved with vision. Vitamin A does a bunch of other things as well. It's involved with some immunity function. It's involving with cell maturation. So when cells divide, vitamin A is going to help ensure that that happens properly. But I'm just worried about the vision component here. So if you know the other one's fantastic, awesome. Okay. Just asking about the vision function. And that's retinol. All right. So when these 2 things come together, they give us our rhodopsin in a rod and what we call general photopsins in the cones. What does that look like? There it is. Okay. Cis retinol. And after light hits it, it changes shape because of a chemical reaction that occurs and changes into what is called transretinol. All right. Vitamin A we get from blood. Okay. So as long as we have a nice diet with a good amount of vitamin A, we can regenerate rhodopsin fairly easy. I don't know if I would ask it as a short answer question or as a process question where I have the steps laid out and you have to put them in the correct order. Okay. That may make maybe an easier question to grade on my part. But the process of changing light into electrical energy is slide 57. Okay. And I'm only looking at rods here. Okay. Let's just be clear on that. We're only talking about rods. Why are we talking just about rods instead of cones as well? Because cones can regenerate really, really fast and rods don't. Okay. It takes us like around 5 minutes to regenerate the photopsins. It can take upwards of like a half hour for rhodopsin to regenerate. And if you've ever gone from a very dark room to a very bright room, as soon as you're in a dark room, right, all of a sudden somebody turns on all the lights and you're like, oh my God, pain. And it just wipes everything out, bleaches everything out. Why does it bleach everything out? Because all that light is causing all of those chemical reactions at one specific time. Okay, so you're burning out all your rhodopsins. Okay, that's what's happening there. So in the dark, rhodopsin is in the cis retinol form. Okay. When light hits it, as you can kind of see right here, see, light hits it, it changes shape. Okay, it changes shape and becomes what is called transretinol. Okay, light hits it and it changes shape. Okay, reconfigures. When it becomes this transretinol, it actually detaches from the protein structure, the opsin. And this is what's called bleaching. Okay. when you bleach rhodopsin, light has hit the photopigment, causes retinol to change shape from cis to trans, and it removes itself from the photopigment. And that's what they're showing right here. Okay. Light hits rhodopsin. Again, very sensitive to light. Causes retinol to change from a cis configuration to a trans configuration. As it does this, it disassociates. It removes itself. Okay. And we refer to this as bleaching. Okay. The bleaching reaction. It takes a little bit of time. Okay. It can take upwards of 30, like I said, but it can take as few as like 17 to 20 minutes, I believe, depending on the book. I think this book says 20. Some other ones say a little bit less. But to get this, to get bent again, requires energy, requires ATP, which is why it does take a little bit of time. Okay. So as long as we stay within a dark environment, this ability to see in that dim light can still happen unless the jokester turns on every single light in the basement and burns all the rhodopsin out, bleaches all the rhodopsin out, okay? Where we cause all of those cis to become trans retinol. Trans retinol removes itself from the opsin protein. And now we have to add a buttload of ATP into the system to try to get all those to come back. Okay? So if the person turned on all the lights in the basement and then shut them all off again, you would not be able to see very well as you did prior to the jokester. Why? You burned out all the opsins. Okay, you burned off all the rhodopsin. Changed all those cis into trans retinol. You utilized all the vitamin A that those rods contained. It takes time because now you got to get the vitamin A out of storage. Got to get it into the blood. It needs a specific carrier. Has to get up there. All this happens relatively quickly. Okay? 20 minutes isn't a whole hell of a lot of time, unless you're my youngest son, which that's forever. But it still takes time because got to trigger a receptor to trigger the body to get vitamin A out of storage and then get it up and transported to the eyeball and then get it in there and cause the ATP to, it takes some time. Okay. Take some time to get all of that to rejuvenate itself. For cones, again, it takes just a few minutes. Okay. I think this book says maybe 5 to 10, I believe. Some other books have a little bit less. 10 minutes is not a whole hell of a lot of time, though. Okay. So that is the basic process that is happening. That's how phototransduction occurs. Light hits these photopigments, rhodopsin in a rod, photopsins in a cone, causes cis retinol in a rod to change into trans retinol. Trans retinol disassociates from the opsin. Need to have vitamin A, need to have ATP. So I can go in there and I can reconvert this back into it. I can add it back to the opsin and ta-da. Okay. I have rejuvenated now this bleached photosynthetic pigment. And you can see again. And all this takes roughly about a half hour, let's say. Let's keep it simple. If it's on a different slide and not slide 57, it's because I probably got rid of a slide, put a picture somewhere else, okay? But wherever it says physiology of vision phototransduction for you, these are the steps for which you can see in the dark. Okay. I may not ask the question, what is the physiology of what happens when you are in a dark environment? I may give you a situation. I may say you are outside trick-or-treating. Okay. It's 8 o'clock at night and you're still trick-or-treating. Somebody shines a flashlight in your face. What is the process that is occurring when the light is shined in your face? And why can't you see that person after they turned off the flashlight? That would be the steps. And the reason why you can't see that person's face very well is because it takes damn 30 minutes to get refilled, to regenerate that rhodopsin. Okay. So I may ask that question as a kind of evaluate and apply what you know type of question. Okay. If I was to ask a visual pathway, I'm going to ask one. I don't know which one I'm going to ask. I haven't finished making this exam at all yet. Where does it all start? In the retina, right? Specifically with the photoreceptors, with the rods and the cones. Okay. We go from bipolar cells to ganglion cells, and it's these ganglion cells that actually form the optic nerve. It's the axons of the ganglion nerves, or the ganglion cells, sorry, that actually form that optic nerve that extends back from the backside of the retina. The great thing about the eyes is that it provides binocular vision. I didn't really go into that at all, but it obviously does, which allows you to see depth, which is fantastic, right? And the great thing about this is that vision images from this side will hit this part of the retina and come back here and stay on this side, and then ultimately back here. Over here in the periphery, as it hits the back of the retina, okay, it'll come here and cross on over. And the same thing happens over here. And then in the back of your brain there at the occipital lobe, we can put all these periphery, okay, coming from this side, coming from this side. We could put it all together and I can create, there it is. There's my depth perception, and I can touch you perfectly fine and not put my fist through your face, right? And that's the great thing about having two eyeballs, okay? It allows that depth perception. It allows you to interact with the world, okay? Again, if you only had one eyeball, you wouldn't have that depth perception, okay? And your ability to interact with the world would be very diminished. But if we look as the optic nerves extend back, here there's what we call the optic nerves, okay? This here. But here it crosses over, and we've referred to this as the optic chiasm. Chiasm just means crossing over. And here is where we get this kind of mixing of the two different fields of vision. Okay. It's basically right in front of the pituitary gland, okay? Right below that hypothalamus. So if you have some tumors associated with that pituitary gland, I mean, it could affect your field of vision. From the optic chiasm, it extends back towards the occipital lobe as what we call the optic tract. Okay. And that's where we get a mixing of the two various different fields of vision. We go to a couple different things. We'll go to the lateral geniculate nucleus, as you can kind of see there, but I can't remember if I changed it over to thalamus. If I was to ask it, that's what I'll ask, okay? Because we did talk about the thalamus. We didn't talk about the lateral geniculate nucleus. As you can see, it goes to a number of other things as well. A pretectal nucleus, which is involved with the pupillary light reflex, which we didn't get into. But the other one I put down here, we did talk about, and that's the superior colliculus. And the superior colliculus was involved with reflexes associated with vision. Okay. So we got the lateral geniculate nucleus. We've got the superior colliculus. Fibers extend back from here. And where do we go? We go to the vision cortex, which we all know is in the occipital lobe. Where does it start? What are the major structures that we run into that we've talked about? Where does it end up? The specific lobe for interpretation, right? All of those structures we've talked about. Those are the ones that would probably be a choice if I was to ask for the visual pathway. Photoreceptors, ganglion cells / optic nerve, optic chiasm, optic tract, to the thalamus or the superior colliculus, and from there back to the vision cortex in your occipital lobe.